In this lecture, we are going to talk about the secondary immune deficiency, also known as acquired immune deficiency. Is the loss of immune function that results from exposure to an external agents. These external agents include various diseases and infections, medical treatment, such as with immune suppressive drugs, following organ transplantation, for example, and social conditions that cause malnutrition. But by far the best known secondary immune deficiency is acquired immune deficiency syndrome, AIDS, which results from infection with the human immune deficiency virus, HIV. One secondary immune deficiency that has been recognized for some time but has an unknown cause is acquired hypogammaglobulinemia. Symptoms include recurrent infection, and the condition typically manifests in young adults who have a very low but detectable level of total immunoglobulin with normal T cell numbers and function. Unlike the similar primary immune deficiency, there is no evidence for genetic transmission of this disease. Another class of secondary immune deficiency called agent-induced immune deficiency, and the example that we are going to talk is AIDS caused by HIV. This is a website. You can watch it, summarize this virus infection. HIV is a single-strand positive sense enveloped RNA retrovirus. That means this virus injects a single strand of RNA into these cells, I mean T helper cells. These RNA must be converted into a DNA using some type of enzymes carried by these viral particles called reverse transcriptase enzyme, converting the RNA of this virus into a DNA. And then these DNA get inside the nucleus of the infected cell and become integrated to the DNA of the host cell. In this case, the virus called provirus or proviral. That's mean the virus, it's ready to be integrated into the host DNA. And we have to know why this virus is pretty sneaky. That's because when the immune cells become activated, they start transcribing and translating protein needs for the immune response. Whenever the immune cells is exposed to something that causes it to start up an immune response, like any infection. But the sneaky part of this virus, this virus needs use this mechanism to transcribe and translate new HIV virus, which bud off from the cell membrane of the infected cell. So, using the internal mechanism of the host cell or the infected cells to produce new viral particles and budding off from these cells. Let us start with HIV entering the body through sexual intercourse, which is how it's typically spread from person to person. The disease begins with acute infection, which is only partly controlled by the host immune response, and advances to chronic progressive infection of peripheral lymphoid tissue. The virus typically enters through mucosal surfaces, epithelial surfaces, and the subsequent event in the infection can be divided into several phases. Here, in the acute phase of infection, which is characterized by the infection of memory T cell, CD4 T cell in the mucosal lymphoid tissue, and death of many infected cells, as well as dendritic cells, which is usually hanging out in the epithelia or mucosal surfaces where the virus entered the body, and capturing these virus and migrating to a regional lymph nodes where a lot of cells, immune cells, are there, which facilitate the attachment of virus to CD4 T cells in these lymph nodes. After the infection of more T helper cells, macrophage, and dendritic cells, this will lead to a big spike in HIV replication and the amount of the virus found in the patient's blood. And the patients will typically experience flu-like symptoms. This is in response to this is the response of the immune system to these viral replications and spread. And the immune system will try to decline the level of the virus dissemination in the body. But the viral will be still detectable by 12 weeks. At this point, the patients will enter a chronic or clinically latency period, which can last between 2 to 10 years. In the chronic phase, the lymph nodes and spleen are site of continuous HIV replication and cell destruction, In this period of the disease, the immune system remains competent at handling most infection with opportunistic microbes, and few or no clinical manifestation of HIV infection are present. Therefore, this phase of HIV disease called latency or clinical latency period. Without antiretroviral treatment, this clinical latency will be ended and the disease will start to end with AIDS, in destruction of lymphoid tissue, depletion of CD4 T cell, and eventually ended with death. In this figure, we can see the typical course of HIV infection in an untreated patient. Soon after infection, viral RNA is detectable in the serum. However, HIV infection is most commonly detected by the presence of anti-HIV antibodies after seroconversion, which normally occurs within 2 months of infection. The viral set point refers to the level of virus in the blood at the time of rebounds, when the immune response began to control virus levels. Clinical symptoms indicative of AIDS generally do not appear for 1 to 20 years after infection, but this interval is variable and is extended by antiretroviral therapy. The onset of clinical AIDS is usually shown by the decrease in the CD4 T cells number below 200 mil and a sharp increase in viral load. Patients become very susceptible to opportunistic infection and other health problems. Typical immune response to HIV in untreated HIV patients showed a rapid increase in viremia in the early acute phase, which declined to a set point. A decline in CD4 T cell associated with the increase in viral load. Okay? HIV-specific CD8 cytotoxic T cell response reduce the viral load. This decrease because the activation of CD8 cytotoxic T cells and associated with increase in CD4 T cell. HIV-specific binding antibody appears after the reduction of viremia, but these antibodies are detectable by ELISA only later in acute infection. In the chronic infection, CD4 cells decline slowly. Viral load remains steady still. Neutralizing antibody begins to appear in this phase, chronic phase. But without a treatment, continued HIV replication, this will lead to exhaustion of the immune system, leading to opportunistic infection and AIDS. This we can see it in AIDS phase, a sharp increase in viral load associated with decrease of CD4 T cells below 200 cell per mil. We have to know why this virus can evade the immune system. There must be a mechanism used by this virus to evade the immune mechanism. We can see it in this slide. HIV has an extremely high mutation rate. Point mutation every day occurs to the epitopes that is presented on these virus and viral infected cells, making these antigens away from the antibody and T cells generated in response to these proteins. HIV-infected cell may evade cytotoxic T lymphocyte through downregulation of class I molecule expression. This way uses cytotoxic T lymphocyte to recognize the virally infected cell and then destruct these virus, killing these virus which harbor killing these virally infected cells to get rid from the viral particles inside these cells. But when these virus downregulate the expression of MHC class I molecules on the infected cell, so cytotoxic T lymphocyte will not be able to recognize these cells. Other mechanisms of inhibiting cell-mediated immunity include inhibition of T helper 1 cytokines and activation of regulatory T cell, which is important in the downregulation of the immune system. So, T helper 1 cytokines is important cytokines to increase the cellular immune response, which is effective in the eradication of viral infection or intracellular pathogens. Downregulation of these cytokines will lead to the reduction in cellular immune response. Activation of regulatory T cells, that means increase in the activity of downregulation of the immune response, because regulatory T lymphocytes are important in the regulation and downregulation of the immune response. And the suppression of dendritic cells function. All these mechanisms used by HIV virus to get away from the immune system. Why the infection with the virus will ultimately end with AIDS and death. The immune deficiency caused by HIV. HIV infection ultimately results in impaired function of both adaptive and innate immune system. As we said in the previous slide, there is reduction in the cell-mediated immunity, which is attributed to several mechanisms. Why this happened? Because there is a direct cytopathic effect and indirect effect. HIV infection ultimately results in impaired function of both adaptive and innate immune system. The most important effects are in cell-mediated immunity that can be attributed to several mechanisms, direct and indirect. In the direct effect, an important cause is the loss of CD4 T cell by virally infected cell. The budding of viral particles may lead to increased plasma membrane permeability and the influx of lethal amount of calcium, which induce apoptosis or osmotic lysis of the cells caused by the influx of water. In addition, HIV infection activates the inflammasome pathway, which lead to apoptosis. While in the indirect damage or mechanism, chronic activation of T cell may predispose the cell to apoptosis, because HIV-specific cytotoxic cells are present in many patients with AIDS, and these cells can kill infected CD4 T cell. Antibody against HIV envelope protein may bind to HIV-infected CD4 T cell and target the cell for antibody-dependent cell-mediated cytotoxicity. This is the way that antibody can target these infected cells. The decrease in CD4 T cell are earliest and most significant in the intestine, leading to the disruption in tissue structure that results in infections with gut pathogens and resulting systemic inflammation. And as HIV infection continues, many aspects of innate and adaptive immunity are affected, leading to AIDS.